the Science Sketches website and go to this section called Create and Submit. And there's a worksheet, this PDF you can download. And the first step is to write a script. You come up with your big question, what you want to explain. The videos have to be less than 2 minutes. That's one of the constraints. And that translates to about 300 words.
So when I storyboard, I'll read the script, and first I'll think of visuals that sort of make sense to go with the script and sketch them out roughly. So that's where storyboarding comes in, where you can flesh out what order you're going to draw stuff so that you don't get disoriented when you're actually doing it. Another thing you want to do is try to have a graphic or a visual representation for each sentence you say in the narrative. So that's also why storyboarding is important, so you can look at each sentence and come up with some kind of visual that makes sense, so you don't have any dead space.
So there are lots of ways that you can set up your camera to film for the sketches, but the main thing is that no matter how you tape it, strap it, or clamp it, you want it to be high enough so that it can see the entire frame of the paper that you're going to be sketching on. And you also want to make sure that it's pretty solid so that any passing vibrations or movements that you make don't affect the image, so it's all wobbly and makes your viewer sick. You also want to make sure that you have adequate lighting. You can use just a simple flashlight on your phone, but you can also go on Amazon or go to a drugstore and find little other lights, or find creative ways to make sure that you have pretty consistent and even lighting throughout the entire plane of field. And one tip I recommend is to have a glue down paper or tape that marks the boundaries of where you can write and where you can be seen once you set up your camera. Because oftentimes you'll be focusing so much on the paper that you'll move your hand in the wrong space or write something out of field. And you always want to make sure that the reader can see what you're doing. And then the other thing is that using your hands is a really great way to connect with the reader in the film, because they can see that it's somebody actually making the video that you're looking at. But you don't want to have your hand and arm all over the place so that you're covering up what you actually want them to see. So remember to write and remove to be able to express what you want to show in your film.
So when you want to go and record audio for the sketch that you make, there's a couple ways that you can go about doing it. If you have any cell phone, the recording device or voice memo app on that totally works fine. You just hold it next to your mouth like you're talking on the phone. And as long as you can get it right in one take, that totally works. You can take that and run with it. Something that we use is this little audio recording device. It's a little more expensive, but if you can get your hands on one, the audio quality is really nice. And then you can process that audio on a variety of different platforms.
When you have all your footage ready to go, all you have to do is import your images, import your audio, sync it all together, and make sure that they look awesome. I'm using iMovie to make this cool stop motion video, which is freely available on all Macs. There's also free software on Windows, and a really cool app called Stop Motion Studio, which you can download as a free app on your phone.
The cell cycle is divided into 4 phases.
Science is only as useful as people can understand it. So hopefully I'm going to be able to take some of these skills and teach them to at least that small portion of people who I know are interested in science communication, and sort of keep on spreading the idea that it's not that difficult to make these little videos, or it's not that difficult to take a minute to explain to people what it is that you do. And seeing results in places around the general public is really what will make people more excited about making these sketches.
Well, our goal is really to get as many people involved in this process as possible. So we intentionally created a really simple format with easy to follow instructions so that anyone can tell a story about science with a video of their own and contribute it to the growing library of videos on our website. All right, let's go back to the slides. Too many windows open. There we go. All right, hand it over to Liam.
Okay. So, here we are. Okay, so that was a big picture overview of the process, and we're going to go into a bit more detail. Just to start with the philosophy and how Lisa and I came to this project. So science sketches, we aim to communicate science using big markers and small words. And so really what we realized was that we both had a passion for trying to create an accessible scientific language to allow people to, sorry, I'm having a problem with the slide advancing. 1 second, I need to re-request.
Want me to go ahead and, okay, there you go.
Okay, cool. Sorry about that. So what we determined was that, from doing public outreach talks ourselves, we realized that many words that we take for granted and concepts that we take for granted are difficult to understand. And this created this awareness on our part of this need, this huge need for stepping stones from a more general vocabulary to the language of science that we all use. So that was one of the things we wanted to address. And we also realized that in talking to many friends and colleagues, that there's a huge desire in the scientific community to contribute to this kind of outreach and communication. But there weren't that many defined opportunities or methods for doing so. And so we tried to find a way of synergizing and addressing these two needs at the same time. And what we thought was, instead of having the small organization try and create this thousands of videos that would explain thousands of concepts, rather we would crowdsource and create a recipe to create videos and have you, the scientists, be the solution.
So our recipe, which you sort of saw in that video that we showed, is pretty straightforward. So the first thing is to create a script. Make sure you simplify it so that it best reaches your intended audience. Then you create a storyboard, images to go with that script, record the video and audio separately, and then edit them together so that you are seeing what is being said out loud at the same time that it happens. So usually that involves, speeding up the video to match the audio bit by bit. And so altogether, especially once you've done this a couple times, it takes maybe 5 hours. The first time is a little bit longer, especially if you're not familiar with video editing, but that's a pretty straightforward process. And really the most important and time-consuming part is getting that script down, figuring out what you want to say and really honing it in a way that best reaches your intended audience. So we have multiple partnerships. The video that we showed you before was from a sketchathon that we held. We've done a few of these live sketchathons, one virtually since the pandemic, but we did a few in person before that, to create videos for this free online textbook called The Explorer's Guide to Biology, which you can find at explorebiology.org. And so we wrote a bunch of scripts ahead of time to define basic scientific terms, and then brought a bunch of people together to get into small groups and just churn out a bunch of videos, which was a really fun process. And that's what you got a glimpse into in that first video that we showed. And then of course we have this really exciting partnership with MBOC, which is promoting scientists to create video abstracts to go along with their manuscript. And what I love about the MBOC effort is that they put the video abstract right there on the same page as the paper, which we'll show you in just a few minutes. And so other people have created video abstracts in the past to put on their own lab website, that kind of thing, which I think is fabulous. And I still encourage that for all papers. But I love that MBOC has really incorporated it into their journal itself. And so it's right there with the paper when you go look at it. So we thought next we would just quickly click around the Science Sketches website. So I've got to switch screens here again. Okay, hold on 1 second. Well, too many things. Sorry, guys. Oh no, I lost it. I lost it because I was full screen. Okay, let me open a new window. Okay, sorry. All right, so this is the Science Sketches website. We have a snapshot of a few different video highlights scrolling through on the front page, but you can find all of our videos here under this All Videos tab. You can see they're organized into different categories. So science fundamentals are really definitions of key basic concepts like what is a protein, what is mitosis, what is DNA, things to give people the vocabulary to be able then to watch more in-depth videos about specific projects. Ongoing research would be you telling the story of what you're doing now, even if it's not at its conclusion yet. So if you just want to tell people what you're studying and why, or perhaps, we are the Holt lab and this is what we study. And that could be the video that people see when they first go to your lab website. Video abstracts, of course, go with specific publications. And then there's a couple of other categories here, science skills, which includes how to edit your science sketch. There's a 2-minute video on that. And then student sketches, which are submitted by students in biology classes all over the world, whose teachers have heard about us, and they submit videos to, go with the lessons that they are learning in class. So if you wanted to find the step-by-step instructions, you would click on this Create and Submit tab, and there's a PDF here. This also exists on the MBOC website, giving the step-by-step instructions on how to specifically submit a video abstract to the MBOC website. But this has everything we're going to talk about today, although we'll take you through it in a bit more detail, and other links that might be helpful. Okay. Now I'm going to go back to sharing the slides. Sorry, I've lost the share button. You would think that all of the Zooms that I've done, I got it back. I'm just trying to get, there we go.
I posted a couple of links in the chat to the MBOC collection of science sketches and also to the Explore Biology website.
You know what? I realized that I shouldn't have started sharing the slides so quickly because the next thing we were going to do was watch another video. So, there we go. All right, so we're going to show you a science sketch that was submitted to go along with an MBOC paper. This is one of our favorites. It involves claymation, so it's 2 minutes, and we hope you enjoy it, and we'll talk about it in more detail.
Cells display proteins at their surface to respond to environmental cues. One example is the insulin receptor. The insulin receptor binds to insulin and then strongly stimulates proliferation and glucose metabolism. Insulin receptors are made by ribosomes that are attached to the membrane of the endoplasmic reticulum, or ER. Newly made insulin receptors fold and dimerize in the ER. They then travel to the cell surface via compartments of the secretory pathway, the Golgi, and the trans-Golgi network, TGN. Cells also continuously remove and degrade insulin receptors at the cell surface. Healthy cells maintain a constant number of insulin receptors at their surface by balancing their addition and removal. In obesity, accumulation of unfolded proteins in the ER causes ER stress. We found that ER stress inhibits the processing of insulin receptors in the ER. Transport of insulin receptors from the ER to the cell surface stops, but degradation continues. This balance of production and removal depletes the receptor at the cell surface, so the response of cells to insulin diminishes. Do unfolded proteins in the ER cause insulin resistance by preventing insulin receptors from traveling from the ER to the cell surface? To find out, we constructed a modified insulin receptor that is made by ribosomes in the cytosol. The modified insulin receptor can be activated by a special drug. We found that unfolded proteins in the ER no longer affected stimulation of proliferation and glucose metabolism when this synthetic cytosolic receptor was activated with our drug. Our work provides a new mechanism for insulin resistance in obesity. ER stress depletes cell surface insulin receptors by blocking their transport from the ER to the cell surface.
Okay, so that was a video created by, ooh, it's still, hold on. Automatic YouTube advancing. Okay. That video was created by Martin Schroeder and Max Brown. And Max actually wrote some music to go in the background. So that was his own composition that you heard in the background, which was pretty neat. There's no need for you to do that for your own video, but if you really wanted to go above and beyond, that's a possibility. Okay, so now let's talk about writing the script, and we'll use Max's video as an example to carry us through.
Okay, so it was briefly mentioned in the big overview video, but in the how to make a science sketches video PDF, one of the first things that we have is a writing worksheet to help you sort of get the big picture and the framework of your scripts together. And so what is the story you want to tell? Who is the audience? So for MBOC, it's a little bit different than a very general what is DNA kind of script. You might be able to use slightly higher level language because you might be talking to a more general scientist audience. But so you need to know who your audience is to determine how to write your script. And then what's the big question and why is this important? So the two things that I have found from editing the MBOC videos so far is that the changes that I tend to suggest the most are to bring out the big picture. Like, what is the really big picture? Why is this question important to ask? How does it fit into, for example, health and medicine, or the big picture understanding of biology? So why is it important? And then furthermore, what's the take-home message? Like, again, you start with a big picture, you say specifically what you did, how you asked the question, and then at the end, you want to zoom out again and say, okay, this is really important generally for this reason. And those are the two things that I tend to emphasize the most. As we said, 2 minutes is about 300 words. So that's a really tight constraint. We speak at about 150 words per minute. And what this means is that you just have to say only what's important. And it's not the same as the abstract that you write for your paper. So we really say, don't start from your abstract. You want to start from scratch. One of the ways that we try to help people simplify their language and become aware of the jargon that is in their everyday scientific lexicon is to use a tool like this one. So we have a link to this tool in our PDF. And you can simply copy and paste your script for your science sketch, and it will highlight every word that is not in the top 10,000 most commonly used English words. So let me show you an example of words that get flagged. So this is a script that was written to convey a message to the general public. And so what's highlighted here in yellow are several words that have been flagged as not commonly used words. And there are a few options when you find words like this. So one option, say, take a term like compressive stress. That's a specific term for physics, but you might be able to change this for the general public and just say pressure. And that is a word that everyone knows more or less what it means. You might make a decision that microbes is a word that is okay. That's up to you. There's also some latitude. There are words that you might need to use technically that are not commonly understood words, but when you have to do that, what we say is that you should, if you don't think your audience is necessarily going to know what the word means, you should define it. So that's done here. In the lower part of the gut, the colon. Just saying the colon alone, you might lose people, but you've defined it ahead of time. So of course, you could do that and use lots of technical language, but don't forget, you only have 300 words. So if you have to define 10 technical terms, you're going to run out of space. So you need to use a combination of strategies to simplify your text and make it as accessible as possible to the people that you want to communicate to. So let's take a quick look at the script from Max and Martin that Lisa just showed. And this is an earlier version of the script. So from the very, very beginning, I think they did a great job of framing the problem at a high level where any biologist could relate to it. Cells display proteins at their surface to respond to environmental cues. And so, they're going to talk about insulin and glucose metabolism. But where we did a fair amount of editing in the end, and we'll come back to this video and you can see how it turned out again, was in these last two phrases, these last two paragraphs. When they first defined their big question, they are talking about like a crux of the paper, a real key experiment. But we thought that we could unpack it and make it more obvious to the reader. So let's look at what they had originally. They say, to show that functions of the ER are required for loss of insulin signaling in ER stress cells, we fused a drug activatable domain to the cytosolic effector domain of the insulin receptor. And then they describe how they do this. And in the end, they say the protein can be activated by the drug AP20187. So we thought that this paragraph, it wasn't obvious what's happening here. It wasn't obvious that we're now entering the point of the story where the key question is being asked. So I'll show how we modified that. And then again, going back to the point that I often try and encourage people to really emphasize the big picture conclusion. This is the conclusion paragraph they had initially. This shows that ER stress cells become unresponsive to insulin because ER stress depletes the cell surface insulin receptors by blocking transport. So again, I changed this with them to really say, this is the conclusion. So here are the two things we're going to look at. And here are the new versions. So in this penultimate paragraph, this is the big question. So let's just say this is the big question. Do unfolded proteins in the ER cause insulin resistance by preventing them from traveling through the ER? So you immediately now know this is the big question. Now we're going to ask it. And then to find out, we constructed a modified synthetic version that doesn't need to traffic through the ER, but rather we can use a drug to circumvent the ER. And instead of using the drug's name, which takes up a lot of time and space, a lot of syllables, we had them refer to this drug as a special drug, which is, it may feel like it's a little simplistic, but it's all you need to know. If you want to know the details, you can look at the paper. And in fact, because we're also drawing, we can add that information directly to the video, and you'll see that. And then that big conclusion, we really just say, okay, our work provides a new mechanism for insulin resistance in obesity. So this is like zooming back out to human health, from the molecular to the organismal. And this is really like a big advance because now, we're putting everything into place. So this is what we found. And this is the mechanism that we discovered. ER stress depletes cell surface insulin receptors by blocking their transport from the ER to the cell surface.
Okay, so next we'll talk about the creation of illustrations and the storyboard. And so I'm just going to go back for one sec to this slide and point out these red numbers that you see in the script. So what you're not seeing on this slide is that below this text, they had, numbers 1 through 18 and wrote out at the beginning, okay, here's what we're going to do for number 1. Here's what's going to show up when you see number 2, et cetera, to sort of map it out for themselves. And so here's another example of that, what the early stages of this might look like for you. So this is a different script, but you can see numbers in pencil on the side there, sort of breaking down the script. And then at the bottom of that same page, this person just drew out, quickly sketched in pencil, what they envisioned happening for each of those steps. And it really is important to have something that you're seeing for every 1 or 2 sentences in the script. So if you have, a couple sentences where nothing is happening, when you watch that in the video, it really feels like dead space. It really feels like the video slows down. So you have to strike this balance between not doing too much and not doing too little. So, you want to sort of show them the right amount to carry them through, and you'll be able to feel that as you watch it in your editing. There's been plenty of videos that I've helped with where, we find a 10-second dead space and we figure something to illustrate for that section that's missing. So you can be really creative with what you include in these videos. So this one, you can see here that it says flip book. And so this person, Jeff Woodruff, created a flip book of cell division and then used sort of that imagery to carry you through to these 3 steps he was going to go into in more detail. So that's something that obviously takes a lot of time. That's up to you if you have that time to invest in the video. But if you do, there's lots of fun ways you can be creative. Max obviously used claymation, which isn't that hard to do stop motion in the end. Lots of people have asked us, how do you do stop motion? And it's really just either a series of photos or, you record the video the whole time, and then in editing, you just remove all the parts where you see your hand moving the object. So that's actually a very achievable thing to do. And you can do a mix of stop motion and drawing, or just do a video that's straight up drawing. And that's also very cool. So then what would you do next? So this is an example of how an illustration might change over time. So another thing that's really helpful is to be intentional about the colors that you use. So colors really helpful in highlighting key messages to carry you throughout the story, and using too many colors can sometimes be overwhelming. So this is a video that I helped illustrate a few years ago, and in the earlier versions, those 2 pieces of paper up at the top, we were trying to figure out how to demonstrate that there were lots of different proteins going into these test tubes. And at first, all the proteins were different colors, and there was a big mass of different colors at the bottom of that tube. And later we decided, okay, just proteins should always be blue. Anytime there's protein showing up in the video, it'll be blue, and people know that blue means protein and ATP is something else. And then also to sort of declutter the page, we showed that, rather than adding things to these tubes separately, we put it together here in the middle, and then showed that the same thing goes into both tubes, and added something else here. This video was also unique in that we overlaid in editing some actual movie data from the paper. So this was a very simple movie showing that egg white alone, when heated up, sort of gets cooked, and egg white plus ATP, when heated up, stays clear. So those were videos that were easy to follow along with, even if you didn't have a scientific expertise in this field. So I do caution that if you do want to include any real scientific data, movies, that kind of thing, that it's something that that people will be able to figure out what it is. So people that aren't in your field might not know that this is the outline of a cell or that kind of thing. So if you do choose to include something like that, make sure that you explain it to the level that everyone will know what they're looking at. Okay, and so then once you figure out what your drawings are going to be for everything, then you figure out what the storyboard or the ultimate layout is going to be. And this is different for every video. So we don't have a set prescription of, your video should be 3 pieces of paper like this one. Some of them are 1 piece of paper, some of them are 6. It really depends on you and what you're drawing and how you're drawing it. One thing that we do caution is that you want to be able to see everything on the page if you were watching this video on a cell phone. So probably you'll be filming this video on your cell phone. So you'll have your phone, pointed directly down at the piece of paper. So as you look in that screen at your piece of paper, make sure you can read everything on the screen. Make sure all the words are legible, that they're not too tiny, that your pen strokes aren't too fine to show up. So if you're trying to cram everything onto 1 piece of paper, make sure that everything is visible. And if not, you might need to sort of scale it up and use multiple pieces of paper. But you do need to think about which images need to go together on the same piece of paper before you change the scene. Okay, so let's go through some of Max's before and after storyboards. So this was the storyboard for his first scene, showing what happens when insulin binds. And what he ended up doing in the actual video was cutting out all of this stuff. You don't really need to see this quite yet, so that you can really focus on what's happening first, all of these molecules of insulin binding to the receptor. And as that happens, he had this really cool effect where the word proliferation and glucose metabolism start off small, and they get bigger and bigger and bigger as more molecules bind. And then this is the next scene. And so because this ended up being a claymation video, it was the same background for both shots. So he didn't end up having to draw all these arrows going back and forth and these X's. Instead, he sort of illustrates this with the pieces moving. So in the normal insulin signaling, you see the receptors being trafficked and moving around and around, whereas in the disease state, you see that they all just get stuck here. There were a couple of things that weren't in the original storyboard that we added. So for instance, in that original script, Liam showed that we added that big question to really set that up before concluding the video. And so now that we've added that sentence, that's a big question, we have to illustrate that somehow. And so sometimes that can be as simple as writing one word really big with a question mark, you know, just something to help focus them that, okay, this is what we're going to talk about now. This is the outstanding question before you move on. So they, you know, had unfolded proteins in the ER. Is this how it maybe works to lead to insulin resistance? And then what I really liked was at the end when Max is making his big concluding statement, he just put two stills side by side of the healthy state and the disease state so that you can look at them and really take it in at the end. Nothing's happening, nothing's moving. You just get to see it one more time as he tells you what he just told you, you know, to wrap it all up. Okay, so I think we're gonna just watch the last 30 seconds of this video again, because it happened a while ago that we showed it to you just to, you know, so you can see again the points that we just made.
Do unfolded proteins in the ER cause insulin resistance by preventing insulin receptors from traveling from the ER to the cell surface? To find out, we constructed a modified insulin receptor that is made by ribosomes in the cytosol. The modified insulin receptor can be activated by a special drug. We found that unfolded proteins in the ER no longer affected stimulation of proliferation and glucose metabolism when this synthetic cytosolic receptor was activated with our drug. Our work provides a new mechanism for insulin resistance in obesity. ER stress depletes cell surface insulin receptors by blocking their transport from the ER to the cell surface.
Okay.
All
right. So, all right, you have your script, you have your illustrations. What happens next? Now it's time to record. We like to stress that you don't need anything fancy for this. You can do everything that we're describing here with your cell phone and with the free video editing software that comes on a lot of computers. So if you have a fancy video camera, great, go ahead and use it. That's fine too. But essentially you just want to figure out a way to point your phone straight down at a piece of paper. So here are a few different example setups. You can see we did have a big tripod here. As was mentioned in that video we showed at the very beginning, it is helpful if possible to have whatever's holding your phone be on a separate surface or separate table than the one that you're actually drawing on, so that you don't get that shaking that will come when your hand hits the table. It doesn't transfer to your phone. So if you can set a tripod on the floor and point it down on your phone that way, that's one way to get around that. In this case, you can see there's a tripod. We had this light box that we taped to it with lab tape. This is a couple different configurations of that. This was a little handheld cell phone clamp that we clamped to a big graduated cylinder that we taped to the table. And this is a lab clamp stand taken from the cold room and wiped down so that it wasn't quite so gross. And there's a little paper towel so it's not actually touching the phone. And that was just a way to, point the phone straight down at the piece of paper. If you get really desperate, you can just hang your phone off the edge of your desk and then draw on the floor. You can decide if you want to have the phone at such a level that the piece of paper fills the frame completely, or it can be higher up and you have space around it and you just trim that down when you're doing your video editing later. So don't be intimidated by this part. You just record everything and then you do the audio separately. So let's go through some key tips. Get feedback at every stage from people who are in your intended audience. So if you're making a video abstract, you want people who aren't in your lab to be able to understand it as well, right? People who might read the paper, who aren't might be scientists, but not scientists in your field. So send the script to a friend who's in a different lab who doesn't study what you study and can read it with fresh eyes. Same with once you've figured out how you generally want to illustrate it, talk that through with someone. Get feedback to make sure that the message is going to land with your intended audience. When you're drawing, draw everything in the correct order. I wrote this twice because it's really important. Draw everything in the correct order. When you're editing the video, if you wrote something in this corner of the page, but you were supposed to see this thing first, and then you draw that thing, there's no way to fix that in video editing. This is always going to be there by the time you show this. So if you realize you've made that mistake, just take that piece of paper away and do it again. I promise it would be more of a headache to try and fix it in editing than it would be to just do it again. On a similar note, make sure that when you're filming, anytime you draw something, move your hand out of the way before you go back and draw the next thing. So it's easy to sort of get focused on what's about to happen. You know, you'll have your storyboard all ready to go next to you. You already drew it out one time. You know exactly how it's going to look. And you have your blank piece of paper that you're filming, and you just draw it all again. And it's easy to just get in the moment and just draw it all out. And then your hand is all over the paper, and and when you're editing, there's no blank space to let people see what you just drew. So be careful when editing that you don't just show yourself drawing something really fast and then move on to the next scene and you're on to the next piece of paper, and the audience never had a chance to sit there for 1 second and take in what you just drew. So it really is, you only need 1 second, 1.5 seconds, something like that, of like a freeze frame on the thing you just drew so that they can look at it and absorb it before you move on to the next thing. But that really requires diligent moving your hand out of the way. So on that note, it helps to work with a partner when you're recording the video. I know this is a little bit more challenging during a pandemic, but if you have a roommate or a partner or someone that lives with you at home, if you're recording this at home, or if you're able to be in the lab and a lab mate can help, you just need someone's eyes to be on the camera while you're actually drawing, so that in case you accidentally lean your head into the frame, they can say, wait, stop, do that again. Or they can help remind you, oh, stop, you just drew something, move your hand out of the way. It just helps to have an extra set of eyes to make everything go more smoothly. When you do record your audio, so I always just do it using the voice memo app on my phone, be aware of background noises. So if you're reading from a piece of paper, don't crinkle the paper nervously. If you're in the lab, be aware of lots of machines and freezers and things that make a humming noise that will get picked up and is very difficult to take out in editing. So a great place to record is in your closet, surrounded by clothes that help dampen the sound. So just be aware of those things when you play it back. As we said, you can use a simple video editing software like iMovie, which comes free on Macs or Windows Movie Maker. We do have a 2-minute video that shows you how to edit your 2-minute video in iMovie on our website that you can watch. And it just breaks down all of the basic steps that are required, which are just, splitting the video up into chunks, speeding it up to match, each thing you're saying as you're saying it. And that's my last point. Make sure they're always seeing what you're saying. So you don't want to be drawing something that you're about to talk about in 10 seconds or vice versa. You want it to line up so that it helps carry them through the whole 2 minutes with you. Okay, and so even though we don't have a tutorial for anything besides iMovie, the concepts are the same. So I can't tell you what the exact buttons are within Windows Movie Maker or something like that, but the concepts of what you do would be the same. Okay, and then here is a bunch of GIFs of some different videos that we created with in those sketchathons that just highlight again how you can really be creative with how you use props and other things like that. So I'll turn it over to Liam to talk about this a little more.
Thanks, Lisa. So yeah, these are obviously really awesome. You can be super creative, you can use claymation, you can use Lego, and we highly encourage it. But on the other hand, I want to make it very clear that you don't have to be a great artist to make a video abstract that's incredibly impactful. In fact, we really try and encourage people who are terrible artists to make these things because they make some of the best videos. And the reason is that art is all about abstraction. And if you want to draw, your science down, like what is in your head, what's your model, then you're necessarily abstracting it. It's much easier to understand a drawing of a brain than a micrograph of a brain, when you've never seen one before. So people who are limited in their artistic ability tend to do a really good job of making sure that they have only what's necessary on the page. So we encourage creativity, but we also encourage inclusivity of people who are artistically not as talented as others, so that don't be held back. So the final thing, we have this fantastic partnership with Molecular Biology of the Cell. We really want to encourage you to get your work out there. It really increases the impact. We'll tweet it out and you get way more views of your paper that way. But if you're interested in general science outreach, we also are super excited about creating better accessibility to science for the public at large. This is a huge issue. We will move the mission of science and we'll create a much more progressive society that understands how to explore the world with the lens of science if they can understand it. And so it's on us as scientists to make sure that we are telling the world what we're doing, explaining to everyone in the public why what we're doing is important, what the big questions are, how they can improve their lives, and getting rid of some of the fear that we see around science. So that's one of the big motivations of Science Sketches as well. And a big part of that has also been our collaboration with Explorer's Guide to Biology. So if you make really great videos, you could be part of that website too. So get in touch and let us know what you want to do, and we'll try and work with you. And so I think that was the end of what we wanted to present formally. Is that right, Lisa?
Yep, that's all the slides.
So we'll open it up to any further questions. I answered a few live, but if you feel like reading them out, it's like speaking them out, or if people have other things that they want to comment or ask, then now is a good time.
Scrolling through the Q&A that I missed.
So the few things that maybe are worth restating. So someone asked if there would be a recording shared of this afterwards, and the answer to that is yes. So you'll be able to go back and look through this. And of course, a lot of what we said you can find on the website. I put the URL to the how-to page in the chat there. Let's see.
There's a new question that just popped up. How do you get over the fear of your research being too specific? So this is also sort of the beautiful thing about making one of these, is that it forces you to think about how to talk about your research to a broader audience. So even if you're doing something that's very specific, there's a good reason that you're doing it, right? And so, it's an important exercise to figure out how to talk about it in a way that is a little bit more broadly accessible. Liam, do you have anything else you want to add to that?
Yeah, I think that that's really a huge part of the point of making these video abstracts when you're submitting to MBOC. Our research is quite specific. We do a really intense study of a particular bleeding edge topic, and it's important to put it in context. And that's what the video abstract should really aim to do. Even though what you might be doing individually is very specific, it's part of a huge jigsaw puzzle that is generally very, very interesting. And that's what we're going to do. We're going to bridge that gap.
Let's see. Do we accept videos made with 2D animation software? Sure. That seems like a lot of work. And if you know how to do that, go for it. Our goal is really just to make it simple so that people who don't feel like they have those kinds of skills feel like they can be involved in something like this too. One thing we do discourage is like straight up PowerPoint, turning PowerPoint slides into it. That feels more like a talk that you might give to scientists. And as the point Liam made, it's easier to include too many details. If you're using something like a PowerPoint slide, it's easier to include stuff that's too high level. Whereas if you're drawing it out, you really are forced to simplify it. So just keep that in mind when you're doing something with, more technical software. But yes, we accept videos like that.
Yeah, so we want things to be artistically driven rather than PowerPoint driven. And the other thing is we really discourage you from including any primary data, because that becomes something that you are blind to. We all know how to look, at least, a lot of people on the call will know how to look at a particular kind of graph or, flow cytometry data, and it makes sense to us. But I remember being a trainee, and the first time I saw a flow cytometry plot, I had no idea what anyone was showing to me. And if they'd spent the time just to draw it out by hand and explain that, you know, this is what you're supposed to be seeing, then I would have understood it. So we don't want you to show a electron micrograph of a, you know, nucleus. Rather, we want you to draw what you're supposed to, what the interpretation is from that. So basically, no PowerPoint and no primary data. It's a sketch. Let's see. So Michelle says that she really likes the idea, is Heidi Walsh's idea, to engage undergraduates to learn communicating science in this format. Yeah, we do too. And we're really excited about partnering with educators. So anyone who is an educator, and it could be undergrads, it could be even high school students. We've worked with people who are, you know, middle school and high school students sporadically here and there. We think that they're at a level, you know, people are pretty technologically sophisticated at a very early age these days. So that's something that we're super excited about.
See, do we have suggestions for holding something like a sketchathon with a class of undergraduates? Yeah, I mean, especially when it's easy to be together in larger groups again. That was something that was really fun, creative exercise for us to do. We did it, I think, 3 times. What was helpful was to have ahead of the sketchathon where everyone's together in person. It was helpful to have those scripts written and ready to go. So with the class, you know, that could be sort of an exercise of, you know, people writing it and sending it to a classmate for revision, that kind of thing. It would be helpful if all that's done ahead of time, and then, you know, a certain number of the top scripts are chosen to go to the next stage, and then you put people into groups of 2 or 3 to then, you know, get together in person and figure out how they want to bring it to life.
Yeah, and there are quite a few resources out there that Lisa has made for how to organize a sketchathon. So obviously, particularly Lisa has done many of these sketchathons all around the world.
Well, workshops, yeah, which is different than like the sketchathon where like a bunch of people come together and make videos all at once. But similar.
Yeah. That's true.
Yeah.
It's similar, but a little bit distinct. But one thing that Lisa did was she made an actual video of how to run a sketchathon, right? Do you have the link to that somewhere?
I don't know what you're talking about. There's an old video of a workshop.
Yeah, there was one that you could step through, and, you know, it was the virtual Lisa that you could have, and it was on a Facebook stream, I think, or something like
that. That was just, I think, a recording of a 90-minute webinar, sort of like this one. But that one is a little bit, so what we're able to do in person, and when it's more interactive, is that worksheet that we showed you at the beginning, we would have people fill that out in the workshop. You know, think about, okay, if you're going to make a video at the end of this workshop, what are you going to make it about? And start writing out those big picture questions and, you know, your take home message, and then get into small groups and discuss it. You know, see, this seems accessible to you, but does the person who studies something else understand what you're saying? And so, especially if it's a big people with diverse scientific interests, I would try and mix them up, you know, put a physicist with a biologist, or, you know, biophysicist with a neurobiologist, and, you know, so that they could make sure they could understand each other. And then later in the workshop, I would have them try and sketch out that big take home message and then do the same exercise again. So a way to sort of start working through that feedback process. And it was always really fun to hear the feedback that people gave each other in person.
I guess we have a lot of questions open, so we should go pretty quickly.
Yeah.
So how many submitted videos make it to your library? Most of them, right?
Most of them, yeah. So yeah, this isn't a problem that we've had that it is a little bit of a problem because this is a passion project for Liam and I. This is not our main job. And so, you know, sometimes it's a problem to like actually get the video up onto the website, because that's something I have to do in my spare time. But yeah, as long as people are open to feedback, you know, sometimes we get videos and then send feedback on how it can be improved. And if you make these changes, then we're happy to accept it to the website. Then it's on that person to decide whether they're willing to make those changes or not. Usually they are, if it's something that they made themselves for fun. Sometimes if it's a student who made it for an assignment, they're not willing to make the changes because they already got the grade for it, they're done. But yeah, as long as people are willing to make those changes. And sometimes they're fine as is, or the changes are really minor editing suggestions to make it easier to watch.
For those student videos that they were not really prepared to update them to a high level, we have a section on the website called student sketches, and some of the more sort of raw ones are collected there. If students are not comfortable at drawing, are there other resources they can use to print out and use them to build a project video? Sure, you can do whatever you want. So as long as it's not PowerPoint or primary data. So you can do things like print something out and sketch it lightly in pencil using tracing paper. And then when you make your video, then you can use a bold Sharpie to make it appear as if you're an amazing artist and drawing a perfect outline of a microscope, but in fact, you're just following a traced line. So you can do whatever tricks you want.
Yeah, we've had people print out and cut out things and then lay them down on a piece of paper. That's also okay, right? If it's a printout of a cartoon of a brain or something like that you feel nervous about drawing yourself, but it's not, like Liam said, primary data or something that would be overly detailed. It's still sort of just a representation, but it saves you the trouble of having to draw it yourself. But then it still has that sort of analog feel of like you're placing it down on the paper and then you can draw an arrow next to it and that kind of thing. We definitely have videos that are like that.
Collage is fine.
Yeah.
Is there any level of scientific curation of the content? Small scale peer review, let's say. Mm-hmm. So yeah, that's a really good question. So far, most of the scripts have fallen pretty close to our expertise, and we feel like we probably would catch just anything that would be crazy. If it were to go outside of our comfort zone, I think we would start to solicit outside advice. Of course, if it's an MBOC abstract, then we can see the paper that it's attached to, and we'll make sure that it's not misstating anything that's in the paper. And that just doesn't really happen. I mean, we haven't got to the level of people sort of, spamming us with conspiracy theories or anything like that yet. So I guess we'll cross that bridge when we come to it. But small scale peer review, that's an interesting idea. And, in the context of an undergraduate class, that would be a fun thing to think about. Could you get the rest of your class to be the peer reviewers for this script? And is it actually true what's being said? So we'll have to figure that out on a case-by-case basis. Okay. Question about fees for receiving your advice. Is consulting with you about video abstracts for free? Lisa, what are your fees?
We don't have fees, but yeah, it's all dependent on our personal availability and bandwidth. So like we said, this is something that is a passion project for both of us. So if you hit us at the right time and we're able to set aside some time for that, we're happy to do that. But yeah,
it's kind of a dream of ours to be able to expand this project at some point. We've flirted with a couple of funding agencies, and hopefully at some point we'll find some funding that will allow us to maybe hire someone like a director for this project, because I'm a full-time research professor at NYU, which is like a 150% job, and Lisa is a full-time communications director, and she has a, how old is her?
I said I have a toddler. My other child.
So both of our time is limited. So we're kind of running at the edge of what we can do right now. But I think that there's so much more that could be done with this project. So if anyone's interested in volunteering, or if anyone has connections to a rich philanthropist, then let us know.
Try and get a bunch of these out. So our collaborations with art students discouraged? No, incredibly encouraged.
Yeah.
Our very first sketchathon, we brought together artists who liked science and sort of artsy inclined scientists. It was really cool. Some of the coolest videos we've made were collaborations with artists. So art student collaborations would be awesome. Are there any examples that aren't molecular, there are things like clinical research, community-based projects, engineering. It's all just who has found us and who has submitted to us. So we are both biologists. So our network tends to be biologist heavy, especially when we were getting started. So there is more of those kinds of videos, but there's at least one clinical research that I can think of. I can't think of any engineering.
There's what is the Higgs boson? That was one that you did?
Yeah. That was sort of a different thing. Yeah. Yeah, so there's a couple of physicsy ones in there, but it's all just who finds us and who submits. We're open to any concepts.
As long as it's science.
Oh, whiteboards. Yes, whiteboards are fine. We use paper because it's sort of easier to manipulate. But these really are, often referred to as whiteboard videos. So sure, you can use a whiteboard. Sometimes there's a reflection. You're asking, are there any downfalls? Sometimes the board itself is kind of reflective, and that can be an issue, whereas paper is not. But it's nice because you can erase, and so, pros and cons.
Yeah, paper is just more accessible, but if you have a whiteboard, then go for it. How can we submit our videos to the Science Sketch website?
There is somewhere on the website it says it goes to like submit@sciencesketches.org, which just goes to my email address. It'll just be me that responds to you. So basically you reach out to me personally, and then we have a back and forth about how you can upload it to any sort of cloud that you may be part of to send me the video, and I can watch it, and then we'll go from there. So you can, if you don't have a way to upload something to your own YouTube channel, we can put it on our own YouTube channel. If it exists on YouTube elsewhere, then we can put it on the Science Sketches website just by embedding that YouTube link. So the MBOC ones, for example, live on the MBOC YouTube channel, but can be embedded on the Science Sketches website. So if your institution has, a YouTube channel that they want to collect that kind of thing, it's best if it lives in one place, and so that all the views accumulate on the same video, and then we can just embed it on the Science Sketches website from anywhere.
Okay. In this competitive world, there are several videos such as Doodly or VideoScribe that may be easier. So what's special about us, and why should you do Science Sketch and not do a Video Maker or Doodly or VideoScribe? Go for it. Like there are different ways you can approach making videos to communicate your research, and I would just encourage you to do whatever method you find the most accessible and easiest. I don't believe that Science Sketches is the only way to go, or necessarily the easiest way to go. It's the way that we found is, I guess we just iteratively tried a bunch of things, and we came to this recipe. And I guess, there are also independent science communication companies where they have amazing video production artists that will produce like very high production quality explanations of concepts in biology or science generally. And that's great. The thing that we wanted to do was to make the process inclusive, so anyone could produce these videos, and that in principle gives us unlimited bandwidth. And though it may take a little bit more time than something that is, in a platform like Doodly, there's no limit to the creativity. So you can use whatever kind of techniques or artistry or three-dimensional stop motion or whatever you want. And so there are no real constraints on the approach you can take. So I think that, but really the bottom line and the answer to the question is, if the way that you're gonna communicate to the rest of the scientific community or the public is through Doodly, then you should definitely do that. The point is just like talk to the world.
There's a question from Undine Cleaver here. Can these video abstracts be used in other journals? Sure. I mean, you can make a video abstract for every paper you've ever written and put it on your own lab website or your own YouTube channel, and we highly encourage that. We love that MBOC has sort of been a trailblazer in incorporating this into its own journal process, where it's really right there with the paper. The only other journal I know of that has done that is Biology Open, just because I was part of making a couple of video abstracts for papers that went there, and we said, hey, can we give you this video to go with it? And they thought that was really cool. And they put it on the website, but they don't have a whole process for it. They were just open to accepting it. Whereas some of the other major journals might not, or they have their own separate process. Like Cell used to have the Cell paper flick, where they might have a different name now, but they would choose one paper per issue that they would invite you to make a video, and they host it. But I love that MBOC has really made this open to any author that's willing to put in the work to do it. So tell other journals to get on board. It's really cool.
I guess the last question, kind of we already addressed. is it okay to use things from Creative Commons, not copyrighted stuff, and include those? Absolutely. Yeah, no worries. So I think that's the last of the questions. And so if you have further questions, then just send us an email. And thank you so much for joining us today. And hope
Thank
to
you
hear from you.
thank you to Ashanti and Teresa from ASCB for helping us handle all this on the back end. And yeah, thank you all for being here.
Okay, bye guys.
Thanks. Thanks so much, Lisa. So you're free to go. I know you have a busy schedule. We'll just capture the chat in the Q&A just so you guys have the questions for your records.
Awesome. Thank you. Really appreciate you both.
All right. Bye. Have a good one.
Bye, you too.
Thank you so much, Lisa. Bye.
Thank you. Bye.
Shanti, I think it's going to stay on live stream until you stop it.